Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Prabesh Bhattarai, an assistant research scientist at Columbia University. Prabesh is also a 2024 Toffler Scholar. Today, we will be discussing how Prabesh integrates single-cell transcriptomics and regenerative biology to advance therapies for neurodegenerative disorders and precision medicine efforts. So welcome, Prabesh.
Thank you so much. It's a pleasure to join this podcast series, and I'm happy to discuss our science.
You have a really interesting focus in your research, and it is really on the regeneration of the body, the brain, the pieces and the parts. I would like to just start with some back projects that you worked, which is, showing that certain fish can regrow neurons that are lost to Alzheimer's. Can you talk about that and really maybe even elaborate on what that means to humans?
Yeah, so probably 10 years back, I started working with zebrafish. It's a small fish that we can in nature we can find along the freshwater river in the Himalayas. And what, let's say 10, 20 years back, people identified that this fish has a special power, like it can regenerate most of its body parts. So in our research, we found it interesting because we want to study how fish can regenerate its brain. But it's a simple question, but how can it regenerate its brain when people like we, human or any other mammal model, they can't regenerate their brain? So it's just a very special question. Even though they are the vertebrate animal, they can still nicely regenerate their brain. So, we in humans, there are complex diseases like Alzheimer's disease where people suffer and there is no cure yet because there are certain things happening in the brain and they cannot recover from it. But in fish, even small injury or even if we create a Alzheimer's disease like condition in them, they can efficiently regenerate their brain. So it was very interesting question for us. So what makes it so special that it can regenerate but we cannot? So we want to understand, if we find out the mechanism how they regenerate their brain, and if this mechanism, if we can implement in mammalian system, so maybe we can also regenerate our brain, and then this could lead us to a potential therapeutic strategy to find a cure for Alzheimer's disease.
And I believe you published on that. You had a team that published. What were some of the big findings around that regenerative, capability?
Yeah, so first we established Alzheimer's-like condition in adult zebrafish, and we see whether they can regenerate or not. So, but we found that they can efficiently regenerate, and we looked into the mechanisms how they can regenerate, or what are the mechanisms that allow them to regenerate. And we found interesting a factor called interleukin-4. It's an inflammatory molecule, and we found that this molecule helps stem cells present in zebrafish brain to efficiently proliferate. So they can sense that something bad is happening in the brain and then utilize this factor which is present in the brain. Like it's an endogenous factor present in the brain. They utilize this factor and activate its stem cells so that the stem cell will produce more neurons so that it replaces the lost neurons, which is the neurons are lost due to the disease, and it replaces the lost neurons so that there is constant turnover of neurons. Whenever there is loss of neurons, there is constant turnover of neurons so that the number of neurons remain similar, or they can efficiently regenerate back to its normal state. and we found this factor called IL-4. It was an important factor, let's say, one of the factors that we identified. And we also tried to check this mechanism, interleukin-4 signaling mechanism in mice, because in mice, we don't see regeneration. So when we checked this signaling mechanism in mice, we found that this mechanism was absent. So probably that's the reason why mice cannot regenerate efficiently in terms of stem cells, say, in the stem cells. So we also tried, so since this was absent, so our next question was, okay, can we induce IL-4 signaling in neural stem cell in mice, and can we see the similar response how we see in zebrafish? But then we found that when we try to activate this signaling in mice brain, but the mice brain, the niche, the environment in the brain did not allow this signaling mechanism to activate. Meaning that whenever we found that the IL-4 acts through IL-4 receptor, which is present in the stem cell, but in mice, its neural stem cell did not have IL-4 receptor. So we did viral transduction to externally express IL-4 receptor in the cell. Naturally, it doesn't happen, but we used a technique to express IL-4 receptor in the cell. But those cells which started expressing IL-4 receptor, they started dying rather than proliferating. In fish, IL-4 receptor is naturally present. It takes IL-4 and then activates the signaling mechanism and promotes regeneration. But in mice, it is not present. But additionally, we try to express this receptor, but instead of proliferating, they rather started dying. So these cells did not survive whenever it expressed IL-4. This shows that the niche in mouse, the neural stem cell niche in mouse, does not support this signaling, but in fish it is naturally happening. So this was one interesting finding, showing that okay, it's not only a mechanism, one mechanism that plays a role in regeneration, but there is a niche, there is a like communication with other cell types probably, which does not allow in mice to this to happen. So then we found another signaling pathway, which is BDNF, NGFR signaling. So that IL-4 was one factor. Then we also looked for another factor which we can activate in mouse, and then we found BDNF. It's a neurotrophic factor, brain-derived neurotrophic factor. It's a well-known factor actually, and we found that this signaling is also activated in zebrafish, and the receptor for BDNF is also present in stem cell population. And when we activate this in zebrafish, it promotes regeneration. And then we try to do same thing in mice, and then we found that okay, neuro BDNF receptor can be expressed in neural stem cell in mice, and they survive. So it was not the case like IL-4, but here they survive, and they also make new neurons, but not to a level where it can fully regenerate and recover. But we found to a certain extent they can recover. And even we did this experiment in mouse model of AD, and we found that amyloid load was also reduced after the activation of this BDNF NGFR signaling. So we also identified these two pathway. And from this, what we found is that okay, not only a signaling mechanism, but the niche inside a brain tissue, like the neural stem cell niche, the environment, the cell communication with each other, everything matters for efficient regeneration to happen. So this was the conclusion that we concluded from our study.
So where's the current state of that kind of research then, especially as it leans towards trying to understand humans and any kind of regenerative capability that humans may have with neurons?
So currently, like in humans, we know that there are endogenous neural stem cells human have, but with age, this number reduced. And in terms of Alzheimer's disease, this number drastically reduced. So there are new neurons constantly forming in humans, but with age, this is reduced. But whenever there is a disease, there is a drastic decrease in adult, like the term is called adult neurogenesis. So the adult neurogenesis is drastically reduced in Alzheimer's disease patient. So this shows that there are new neurons like forming, there are few neurons dying, there are new neurons forming, but whenever there is disease condition, the number of new neurons is not up to a level, but number of neurons dying is very high. So that's why there is a big gap, and eventually in disease like Alzheimer's disease condition, the patient, they lose their neurons progressively, and that's why the disease happens. And to like current research to activate adult neurogenesis or to start regenerating in human brain, it's a bit far-fetched, because like naturally there is already a decline, so we need to additionally add up to compensate the neuronal loss, but which is not the case so far. So what now we are doing is we need to find a stage where we can already start training our cells to produce new neurons at early stage, not during a disease condition, but a very early stage. But it's very difficult to say at current scenario when should we start, and like how this approach will lead to, because we are still figuring out how we can enhance the regenerative response in mammalian brain. So we first need to check all these things in mammalian models rather than to go into humans. It would be very it's a long road. But what we can do is like there are different aspects of research. So we need to fully understand the mechanism of a disease, how disease is happening, and at what rate and at what stage the different cell types are being affected. So maybe neural stem cells are being affected way earlier than the disease is being diagnosed. So that's why we need to find a point where before a disease is diagnosed in human, probably neural stem cells are already under stress. Like it is already being affected due to a different microenvironment in the brain, which we need to identify first. And if we can figure it out when to start to train our neural stem cells, endogenous neural stem cell, that eventually these neural stem cells will be lost. Eventually, there will be a lot of neurons dying whenever there is disease. So we need to train these neural stem cells to be to activate way earlier than it is late. It could be too late when we diagnose Alzheimer's disease. It could be too late for the remaining endogenous neural stem cells to activate and proliferate and produce neurons. So we need to start this way earlier. And I don't think only this approach will help, because Alzheimer's disease as such is a very complex disease. So it's not only a disease where neural stem cells are affected. So there are other, like neurons is affected obviously, but there are other cells like glial cells also act very differently, and neural stem cells are a subset of this glial cell. But other glial cells might also interfere with this small subset of glial cell which acts as neural stem cells. So we need to understand every aspect. So every factors which are affected in the disease. And currently, like the field is very opening up. People were only studying neurons in Alzheimer's disease, but more recently they are studying neural stem cells. They are studying like other cell types in the brain, like for example microglia, which is like immune cells in the brain. And like we are also looking into extracellular matrix, so ECM components in the brain. So these are also being affected, and that's why we are looking into gliovascular unit or blood-brain barrier. So blood-brain barrier is also one important aspect where, due to Alzheimer's disease, there is leakage of blood-brain barrier.
So tell me a little bit, or tell us a little bit about kind of your current research. Because of this complexity, there's a lot of different places where you can focus. Where is your current focus right now in your research?
So we are currently checking blood-brain barrier, how blood-brain barrier is affected in Alzheimer's disease, because we know that there are various genes or there are various factors that are known to be risk factors for Alzheimer's disease, but we don't know how this particular gene XYZ is a risk factor. So we need to understand the mechanism behind these risk factors. And recently we picked some risk factors which are known to be also risk factors for cardiovascular disease, for example. And we know that Alzheimer's disease and cardiovascular disease, they often coexist. And one common factor would be, so these genes or these factors, they're affecting the disease by means of blood-brain barrier pathology. So, because in brain, it is also highly vascularized tissue. So there might be some phenotype or some things happening that is also contributing to leading to Alzheimer's disease. So, and it could be also one of the initial event that eventually builds up and leads to Alzheimer's disease. And one crucial cell type which is present in the blood-brain barrier is also glial cells. So, which a subset of glial cell is also neural stem cell, and a subset of glial cells are also present in the blood-brain barrier, and it's called like gliovascular niche, where there is a blood vessel around there. There is a glial cells. There are endothelial cells. There are pericytes. There are microglia around it. So in this aspect, so we want to study how, or like glial cells, how cells are contributing to disease pathology, be it in terms of reduced neurogenic potential, like where stem cells are reduced and so on, or in terms of gliovascular unit, how like glial cells are contributing to maintaining the blood-brain barrier, or like what results in disruption of this blood-brain barrier. And we are mainly studying role of glial cells or astrocytes, let's say.
And you, to do that, you use some pretty advanced technology. I believe this single cell transcriptomics, whatever that may be, if you can explain what that is and give us a visual of the complexity and what that really kind of opens up as far as understanding.
We use single cell transcriptomics. Transcriptomics means a transcriptome level in a single cell level. So how each cell's transcriptome means the RNA expression level. So like we could go to a single cell level. So earlier bulk transcriptomics was heavily used, but now with advancement there is now single cell transcriptomics. So because we could see in each cell, like how one glial cell is affected in disease condition compared to how that particular glial cell is in like healthy condition. So we could see what is the difference. And since there are so many cell types in the brain, in bulk transcriptomics, everything would come up. So whatever effect we are seeing in glial cells could be normalized because of a lot of neurons being there, for example. So, or how a microglial contribution like a microglial cell, its contribution would be normalized because there is so many glial cells or neurons coming up. So we can't differentiate the small changes. So with single cell, we can separate microglial cells only and see and do the comparison and see what is changing. Or we could only look into glial cells and then see what are the changes. But what single cell further does is that even within glial cells, there are different subtypes of glial cells. Certain glial cells, which we can say reactive astrocytes, let's say, these are some subset of astrocytes which is activated because of disease condition. And there could be another subset of astrocyte which could be neural stem cell, and these are reduced during disease condition. So, and these are the subtypes which, like, for example, a subset of astrocyte which act as neural stem cells. These are the group of population that we want to train and we want to activate more so that it can contribute towards regeneration. Or there could be another subtype of astrocyte which is present around the blood-brain barrier. So, how they control the different mechanism to maintain the blood-brain barrier. But in disease condition, there could be some mechanism that is disturbed, for example, in these astrocytes, and this leads to blood-brain barrier disruption in disease. But in healthy, we could also do the similar single-cell transcriptome profiling in healthy brains, and we could see that this subset of astrocytes which are present in the blood-brain barrier, the mechanism is intact. Everything is fine, and that's why the blood-brain barrier is also intact. It's functional. But in disease condition, there might be this small factors, some mechanisms that could go wrong. So we could pinpoint, oh, the astrocytes which are present in blood-brain barrier, these mechanisms, XYZ mechanism is wrong, like this is disrupted. That's why it is disrupting the blood-brain barrier, and that's why it is adding up to a disease phenotype. And maybe this could be a sequence that eventually adds up and leads to Alzheimer's disease. So single cell transcriptomics have helped a lot. And now there is another advancement. It's called spatial single cell transcriptomics, which means in which part of the brain, which region of the brain that these cells come from, we could also identify that. So for our blood-brain barrier pathology to investigate this, so if we do spatial single-cell transcriptomics, we can also pick, oh, these are the astrocytes which are present in blood-brain barrier. So we could also visualize spatially. So this would be a new direction. Like now, spatial transcriptomics is also getting very famous in our research, and like we would also like to use that.
How do you keep it all straight? There's so many variables. There's so many, well, it could be this, it could be that. We see this, we see that. How do you keep it straight as a researcher and stay patient and focused, but yet still be exploring those what ifs?
Yeah. So as a researcher, we should be very patient because like there is so much to know. Like it's an open field. There's so much to know, and the more we dig in, we feel that okay, probably there is even more to look into. And whenever we study, so we only look into few variables because we don't want to open everything, and we always compare it with the control groups. So when we check, for example, a subset of astrocyte which could be present around the blood-brain barrier, so when we look into the control, there could be numerous genes that could be changing, but we look in there could be already some studies which is published that okay, this factor has already been shown to support blood brain barrier, let's say, like, and another factor is already it's been shown that these are expressed by astrocytes. And if we see such factors, we'll have list of candidate genes, and we'll look into those particular genes. We'll look into the literature, what other people have published, what other people have shown, and based on that we try to fill up the gaps and see whether it would make sense or not. Because analyzing this transcriptomics data is also very complex. We need to train, like, it's a huge amount of data. We need to pick properly. We need to analyze in such a way that we reduce this number of candidate genes. And looking into those candidate genes, then looking into a literature about those candidate genes, and we come and pick up a few genes that we want to test. And when we test, we also have to do a lot of experiments, whether we create a hypothesis that, okay, factor X is involved in this mechanism which maintains the blood brain barrier. And then if we find this X to, gene X to be absent in terms of, it could be two ways. So in disease condition, certain mechanism is upregulated, and in healthy condition, that mechanism is downregulated. So we know that, okay, having this mechanism could be a marker for a disease condition. Or it could be that in a disease condition, like factor Y, let's say, a mechanism of factor Y, this is downregulated. But in healthy condition, this is up. So this shows that this factor is essential. So it's a beneficial factor which we lose during disease condition, and it could be because of this factor why we are observing this disease phenotype. So yeah, we need to study, we need to look into every aspect, and we need to do a lot of literature review where whether this factor has been shown previously, whether it is involved in this disease context, and so on. So it's a laborious process. We need to study properly.
And right now, you use primarily mouse models, or has your lab moved into like ISPCs, or where are you, kind of at with the tissue?
So we use geographies as a model because we want to study like regenerative aspect. We also want to study blood-brain barrier pathology, and geographies is a useful model. And whatever findings we find in geographies, we try to validate also in mice and also in iPSC cells in 3D high result culture using either primary astrocytes or also using iPSC models. So we do like cross species validation of whatever findings we have. So now, like currently, we are looking into different risk factor genes for Alzheimer's disease, how they lead to Alzheimer's disease. And mostly the factors which we are looking into has a role in astrocytes, or they're expressed by astrocytes, or these factors affects the astrocytes, whether it's a ligand produced by astrocytes or whether it's a receptor that astrocyte has, and the ligand binds to the receptor present in the astrocytes and the downstream signaling. So it is mostly revolving around astrocytes because it's an important cell type, because it's also part of the blood-brain barrier, plus it is also a subset of astrocyte is also neural stem cell. They act as neural stem cells. So our focus would be to have this both aspect together, and that's why we look mostly into astrocytes.
And that's kind of a new area of study. Is that correct? That
Yeah, so even neural stem cells and regeneration is also a relatively new area, like probably 10 years or so. People have been looking into this, and also blood-brain barrier pathology in Alzheimer's disease is also relatively new. So what we found more recently was we identified fibronectin 1. It's a ECM extracellular matrix component. We found that this fibronectin 1 is found to be highly expressed around the basement membrane of the blood brain barrier. So there is increased deposition of fibronectin 1, which thickens the blood brain barrier part, and this leads to activation of or disruption of several other mechanisms, signaling mechanisms and signaling molecule that is known to be protecting the blood brain barrier or maintaining the blood brain barrier. But since fibronectin is depositing so much, there is disruption of this, a signaling pathway, and this could be one of the reasons why we see blood-brain barrier pathology and disruption in Alzheimer's in the course of developing Alzheimer's disease. And fibronectin 1, we know that this is secreted or expressed by astrocyte population and secreted in the ECM region. And probably this could be one of the events that we could look into, whether reducing fibronectin 1 could be one potential therapeutic approach that we could check. And what we also identified was when we looked into whole genome sequencing of some some patient, where that the data that we have in Columbia University, we found that there is risk allele gene ApoE3 and ApoE4. So in Alzheimer's disease, ApoE4 is known to be one of the strongest genetic risk factor. So we found that if there is a variant in ApoE in fibronectin 1 gene, and if this variant is present in ApoE4 individuals, it's most likely to provide protection against Alzheimer's disease in these ApoE4 individuals. So this showed that okay, fibronectin 1 could be one pathological marker or pathological feature that could be targeted to protect against Alzheimer's disease, because a variant which is known to be reducing fibronectin 1 is directly linked with people not developing Alzheimer's disease, even though they have ApoE4, which most of the like 80% of the people, if they have ApoE4, most likely they will develop Alzheimer's disease. But even though they have ApoE4, just because of presence of this variant, it showed that okay, they are being protected against developing the disease. And why is it the case? Probably because of this variant, which is known to reduce the expression of fibronectin 1. So there is less fibronectin, and we see that even we did validation using postmortem brain tissue from ApoE4 individuals who don't have Alzheimer's disease, we see that, oh, there is reduced fibronectin 1. So it could be one potential mechanism that we could look into. And now we are studying more to dig into, okay, how fibronectin 1 is involved in the disease pathology. So this is one fact. And we could also check in terms of a neural stem cell perspective, like whether fibronectin 1 has a role into neural stem cell or subset of astrocytes, if they also have a role. So that we have one factor which is contributing to both the aspects. So since I told you earlier that Alzheimer's disease is not a one fact, it's a multifactorial disease, and it's better to look into different aspects rather than just focusing on one. So we are trying to explore different aspects. So two of the aspects that I am interested and I'm particularly doing is neural stem cell and regeneration aspect plus blood-brain barrier pathology, where currently we are studying.
Yeah. Is there is there any relationship with this, with ApoE2? Because isn't ApoE2 kind of a resistant? Have you seen any kind of connection there?
Yeah, so ApoE2 it's a very rare population. So we don't have ApoE2 samples yet. So we have not checked, because the ApoE4 is actually an endogenous, or the natural allele, and it got modified, or it got mutated to ApoE3 allele, and then it got mutated to ApoE2. But ApoE4 as such was the main, or the first allele. And if you see globally, ApoE3 is very abundant. Probably 80% of the global population is ApoE3, and then ApoE4 is around 10 to 12%, I think, and ApoE2 is the least one. So we have focused more into ApoE3 versus ApoE4, but we have not looked into ApoE2.
Yeah, I was just kind of curious on that. Looking at this regenerative kind of capability, the way I'm understanding what you're saying is you've got to understand kind of the mechanism for the disease and how that's all kind of happening to be able to go back to understand, where do you stop it? Where
Yes,
is
interfere
that
or
pivot point?
yeah. So because it's a chronic disease, we might be when doctors, or when they see patient, probably disease is being diagnosed at one particular point, but maybe the molecular mechanism how the disease progression starts, it could be way ahead in time than when the disease is being diagnosed. So, and lot of events in different aspects, be it changes in ECM component, be it changes in neural stem cell aspect, be it changes in microglial dysfunction, or be it different aspect of a disease. Maybe several events are already starting to go wrong at the very beginning, and maybe we need to understand the mechanism in these area, or at this time point, so that we can already interfere at this time point. So there could be several mechanisms which we can call disease modify or disease causing mechanisms. So a lot of disease causing mechanisms can already go wrong at way earlier in a molecular level, and we are only being able to diagnose the disease at the later stage where it is too late. So that's why understanding the disease progression at an early event is essential. And while studying the disease progression, look into the mechanisms which causes the disease, and maybe find a target or find a molecule which could alter this mechanism so that the progression is stopped. So this is where I think we have to focus, and we are focusing at that. So that's why we are picking this risk factor genes for Alzheimer's disease, and we are trying to understand how the disease progression happens in Alzheimer's disease. So if we know the mechanism, maybe we will also know where to interfere, and with what mechanisms are causing or leading to progression of disease. That's why we can interfere with certain molecules, small molecules, or modify these targets and then stop the progression of disease. And even though if we are not able to stop, we can probably delay the progression of a disease. So that could already help eventually, because with fibronectin 1, we know that if this variant is present in ApoE4 individual, so the disease is delayed by, I think, 3.5 years. Onset of a disease is delayed by 3.5 years, which is like 3.5 years, just because of this one particular variant. So if we can understand the mechanism further, maybe we can prolong this period, or we can even stop like the progression. So, but this is where we want to, like, we are focusing our one of the research is focusing in this aspect.
So I think there's, it sounds like you need better diagnostics. We're going to need to have maybe some sort of testing for this. Is that like a blood test or some sort of test, as well as the therapeutics? How not to put you on the spot, but I mean, is this 20 years away? Is this 5 years away? Where do you think when you just see the acceleration of the knowledge that we're starting to gain, where do you think this is going to all kind of come to play?
It's very difficult to say that, because what in the field we are lacking probably is the longitudinal study, for example. So we only know this in animal models. Like, we can study the progression of disease in animal models properly, but in humans, maybe like we cannot see that. But probably where from the patient, if we can get like genomic, like blood samples, and from there we can take plasma, and we can study in plasma different, we can do proteomics, like it's another omics approach. We can see what are the things that is changing in time. So, but for this we need a lot of volunteers, where we, like, to study properly, and we can see what changes, and maybe from there we could find some biomarkers, or we could also find some factors which could be either a causative of a disease, or it could be as a result of a disease. So either way, it could be used as a biomarker, and then we can study whether this factor is involved in progression of a disease. If this factor is involved in a progression of a disease, then we can try to interfere the mechanism how this factor is responsible, and like that could be a way that we could proceed. And in how many years that we could achieve this, it's very difficult to say, because it's not only one or two experiments that we can do. We have to do several experiments where, and at a different level. So we need to do like basic study using, like in cell culture or in fish model or in mice model. Then we need to do a proper preclinical study, where in large number of animal models we need to test, and whether it's successful or not. So, and then the next step would be, because we are doing basic science for now, but in like to move into translational research, it's a bit like it will take some time in this regard. Like with fibronectin 1, we are studying extensively regarding the mechanism and so on. So we know a mechanism, and we are also looking into some compounds which could be useful to mimic like similar scenario, and we have to test in a lot of preclinical models. Then only we could go ahead. So, but yeah, so it's an open field. It's a complex disease that we are looking into, and but there has been some good progress. And it's a collective effort, so it's not only us. There are different groups which are studying into different aspects, maybe combining all the aspects and having one, working together in this field to understand the disease better, and then to find the therapeutic targets better, and then to tackle the disease.
So, as we kind of wrap up here, what do you on that is very complicated, and you won't be able to forecast the time, and I appreciate that, and I understand that. What do you think is going to be a big game changer or breakthrough to kind of break the dam a little bit, if you would, for some of this information that you're talking about that we need. Do you have any ideas of what that might be?
So it could be the biomarker or early detection of a disease. Now we like the disease is being diagnosed probably a bit later, where it's probably a bit too late. Maybe early disease biomarker, like if we can find out that, then it could be a like very valuable information, because we know when to interfere, like from that point on, when to interfere to make neurons bit resilient, or to activate neural stem cells to produce more neurons and regenerate and neurogenesis, and also like different aspects of the disease. So where we can like boost our immune cells, where we can make neurons a bit more resilient towards the upcoming events. If we know the early biomarker, then maybe to those individuals who we know that will have a disease, maybe we can already start treatment at an earlier time points, at earlier stage. So probably this will help. And also like if we maintain our lifestyles and so on, it will already help, like having a lot of physical exercise, having a proper diet, and all these things. So like having proper sleep and more exercise, proper sleep, like avoid bad habits like smoking and so on. So these are all environmental risk factors which contribute to disease progression. So maybe like this aspect together with the treatment option, that where we can interfere with the disease progression at an early event, maybe this together will help delay the disease, or even avoid developing the disease. So it could be that. I think we need to more look closely what are the early events, the mechanisms of the disease progression at earlier stage, and maybe find a biomarker which detects the disease at an early event, and maybe so that's the way to go, I think.
Yeah, no, I agree. It's such a horrible, disease, right? Alzheimer's, any of the dementias, just they're horrible. And if they are happening sooner, it would be good to stop that, because just really bad. So, this has been wonderful, and I've learned a lot, as I do when we chat. Just kind of finally here, how do people get in contact with you if they want to find out more of what you're doing, or fund you, maybe start this longitudinal study that is so important? How do people get a hold of you?
So I'm an associate research scientist in Columbia University in Taub Institute. Yeah, I can provide my email, and I am happy to take questions or advice even, and also like happy if somebody is willing to fund. I'm happy to do that. And I have been funded by Toffler grant, and I have used this grant to do a transcriptomic study actually. So it was very useful. Now we are working on preparing a result for this, and we'll hopefully have some publication out of the research that we are doing. So yeah, so I can provide email. So my email is pb, like P as in Prabesh, B as in Bhattarai, 2886 is my uni code, at cumc.columbia.edu. So in this, you can write me email, and I'm happy. I can answer any questions, or I could also give further insights what the research we are doing, and what are our next steps, what is the direction that we want to go.
This has been wonderful. Thank you so much for your time. And I know you've got an experiment that you're doing right now, so you need to rush off to that. But thank you so much, Prabesh.
Yeah, thank you so much. So it was nice discussing about my work and my future plans, or my ideas, what we want to do, or what is the state of the field, and so on. So it's been a nice experience. So thank you for the opportunity.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.